Hi everyone, my name is Madison Kalb, and today I'll be talking about circular permutation. So first things first, what is circular permutation? Circular permutation is the reorganization of a protein by changing the location of the C and N termini through fusing the original termini together and creating new ones. In doing so, structurally similar proteins with altered amino acid sequences are created. So here's a simplified version of a protein with 3 chains. I'll call them X, Y, and Z. In this protein, the N terminus is at the beginning of chain X, and the C terminus is at the end of chain Z. So the overall sequence of the chains is X, Y, and Z. But let's say a circular permutation occurs, fusing the N and C termini together and creating a new termini between Z and Y. The new protein chain sequence is now ZXY. So structurally, these proteins are very similar, but the sequence is in a different order. Okay, so now we know what a circular permutation is, but the next question is, how do they occur? Circular permutation can occur naturally as a result of evolutionary events like gene duplication and fission or fusion, or without genetic changes through post-translational modification. In gene duplication, a gene encoding a protein is subsequently duplicated in the genome. Using our XYZ example from earlier, this would result in the sequence XYZ-XYZ. A later introduction of start and stop codons within the repeated genes removes the redundancy in the protein while also rearranging the amino acid sequence, creating the sequence ZXY. Fission and fusion occurs when partial polypeptide sequences combine to form a single protein. Circular permutations can be generated in the lab using restriction enzymes to cleave and ligate the gene at particular locations or using PCR-based cloning methods. In PCR-based cloning, an oligonucleotide encoding both the N and C termini and a polypeptide linker is used to fuse the gene in one place, and new termini are generated by either selectively or randomly cleaving the DNA. So what are the advantages of using circular permutations in protein engineering? The addition of circular permutations can provide information about the folding order and role of specific structural features in the original protein. They can also be used to change properties like reduce proteolysis by protecting the C and N termini, improve catalytic rates, increase stability, and alter substrate or ligand specificity. In this study, circular permutation was used to broaden and increase the catalytic activity of a lipase B. Random permutants were created using PCR-based cloning and DNase I to randomly cleave the gene. One variant introduced new termini to regions near the active site and resulted in catalytic activity that was increased and increased specificity for bulky substrates. This change is a good example of the ability to use circular permutation as a tool to change active site access and alter flexibility of specific regions in a protein. Alright, well that wraps up this synopsis of circular permutation. Thanks for watching.